Good evening. Hi, Dr. Kapil Gupta. I am here to present the my teaching concept, agrobacterium mediated plant transformation. So first, we would like to know what is agrobacterium. Basically, agrobacterium is a gram-negative bacteria that is also known as a natural genetic engineer because it engineer the host DNA and make changes according to its requirement. So that is why it is known as natural genetic engineering tool. And it is further used in various application where we need to modify the genome or genomic sequences of a particular process. So it is a plant specific and it interacts with the plant cells or the tissue or the plant. It transfers because agrobacterium tumefaciens it interacts with the plants and it infect to make a tumor-like structure that is say you can say that is known as a gall tumor or crown gall diseases that is caused by this agrobacterium tumefaciens bacterium where it transfers a DNA that is called a tDNA because this DNA is being transferred and that is why it is called tDNA. And this tDNA is transferred by agrobacterium to mutations to the host genomic DNA. So we can see here there are several pictures that shows that these crown gall or tumors has been arose due to the infection of this agrobacterium tumefaciens. It can be very small up to a very big up to few meters large enough. So this picture represents this process of DNA transfer by the agrobacterium to the nucleus of the plant cell. So here you can see a pipe-like tunnel or a channel is generated where a copy of DNA that arise from the tDNA or Ti plasmid is being translocated to the nucleus of the plant cell and where it gets integrated. So this is DNA transfer process and an integration of this tDNA makes stable changes in the plant genome. So we are discussing about the Ti plasmid. What is this plasmid? Basically, inducing yeah, or tumor induction that is mainly occurs due to the Ti plasmid. This plasmid basically is responsible for the tumor induction, and that is why it is Ti plasmid, where it carries several features by which it creates a tumor-like structure and that are known as oncogenic genes. Here you can see the oncogenic genes. They are basically auxin and cytokinin producing genes that makes different enzymes that synthesize these phytohormones and they start the division of the cells of the plant very rapid manner so that the cells divide in an uncontrolled fashion and they make a tumor or protuberance coming from the stem or the root so that makes a tumor like structure. It also synthesizes some opines because this plasmid uh, opine synthesizing genes they are required by the agrobacterium to synthesize opine because it grows on plant due to this opine where it utilizes this opine as its nutritional source. Here, this tDNA region that is transferred to the plant is limited by a left border region and the right tDNA border region and only this left border to right border region is being transferred and none other than this region can be transferred to the plant. This is the beauty of this system. So there are additional genes like opine catabolizing means have those genes or those protein coding genes that are required for metabolizing this organic compound opine is synthesized by this agrobacterium Ti plasmid so that agrobacterium utilizes the synthesized opine for its growth and the other virulence region, that virulence region basically responsible for creating this infection and This virulence region basically it has six Vir genes, Vir A, B, D, E, F, and G, and they work in a operon manner and they are very synergistically acting to start the process of transfer up to the integration of this DNA to the host genome. So what are the steps are involved? What in the process of the DNA transfer through the agrobacterium? First, signal transduction means the signal comes to the agrobacterium through the wound and a phenolic compound is sensed by agrobacteriums, then it starts signaling, so it makes its things ready. And second, attachment of agrobacterium to the plant cell. So it through the process of chemotaxis, this agrobacterium travels toward the wounded plant cell or the cells which secrete some sugar or phenolic compound so that it moves toward that cell and attaches to that site. After attachment, production of virulence proteins is started by the virulence gene, Vir genes. And after successful production of the Vir genes, the tDNA strand is formed in a single stranded form. After that single strand tDNA is formed, it starts transfer. And after the transfer and reaching into the cell, plant cell, it integrates into the genome. So we just like to refer to this picture where a plant cell and a bacterium cell, they are shown together to inform us that how this all process starts, because when any wound occurs or any wound condition, there are some compounds like phenolics, some sugar and some acids are secreted. So after synthesis of this compound, the if any agrobacterium sense it, it reaches to that site where this wound is present. And after the sensing, the signaling starts where these phenolic or sensory compound signal compounds start synthesis of Vir A. Vir A further activates Vir G, and, and this Vir G is a activating or it has also autocatalytic activity. It activates other Virg expression, and further, where Virg expression or the proteins are in sufficient amount, they activates other Vir genes like A, B, D, E, F. So when these virulence proteins are synthesized, they start formation of single strand of the transfer DNA, and after the transfer, this system makes a secretory system, tunnel-like structure from where the single stranded DNA is translocated to the plant cell, and from where the single-stranded binding proteins carries or protects it from degradation of the host machinery of the plant, and further they are moved to the nucleus, and in the nucleus in the chromosome it goes and integrates, and further it makes being double stranded, it successfully integrates in the chromosome of a host cell, and after integrating it becomes a part and it is carried forward to the next generation to, or with the cell division it is carried or it is transferred to the other cells with chromosome as well. So it is all the process from where a signaling starts and the tDNA is just made in a single stranded way and further transferred to the plant cell where it integrates and makes a successful integration, and then the due to this tDNA, the agrobacterium synthesizes required compound and it starts it growing, and further and excessive growth occurs and there is a coming or bulging of the tissues, so that makes it a tumor like appearance. Thank you very much. Thank you.